So hello everyone. So today we are privileged to be joined by Dr. Christine Wangia. As I've said, I'm my lecturer, and Dr. Christine Wangia is a pharmacist by profession and a researcher in medicinal plants. She holds a Bachelor of Pharmacy and MSc in Pharmacology and a PhD in Phytochemistry. She's a senior lecturer in the School of Pharmacology and Pharmacognosy at Jomo Kenyatta University of Agriculture and Technology. And she's also an entrepreneur with over 30 years of experience. I'm sure we shared a standard article that had circulated some time before. So I'm sure that some of you saw the article featuring Dr. Wangia's work. And she discovered a herbal remedy that has anti-arthritic, analgesic, and inflammatory properties and can be used to treat rheumatoid arthritis. So thank you so much, Dr. Wangia, and welcome to the session.
Thank you, Daniel, for that humble introduction. I think, can I share my my slides? Is there not an opportunity to share?
Yeah. You can go ahead and share.
Okay, I've seen it. So first of all, I'm really excited. As I've been introduced, I'm Dr. Christine Wangia. Daniel is my student, and I was really humbled by him and your group, SAS Africa, that I can share my general information on drug research and discovery. And Daniel told me not to go into the details of the technical aspects, but as pharmacy students or medical students, at least I will share a little bit. It can't be general completely. So I'll try to do to share as much as possible the general aspects, but I think from that you'll be able to get something. So actually my studies on Ruellia species, the plants that I work on are called Ruellia species, and they are perennial creepers. They belong to the Acanthaceae family. So these are the macromorphological features of Ruellia prostrata. I worked on 3 Ruellia species for my PhD. They had similar activities, and further work was on the one that was the most potent. Otherwise, they had similar activities, but Ruellia prostrata is the one that was most active. So most of the work I continued with the Ruellia prostrata, but for my PhD, I worked on 3 Ruellia species. So this is the maybe I can hide. This one shows the root system. The roots go down into the soil up to even 4 feet, and they are small creepers. This is the vegetative and root system. These are the flowers. These are the fruits. This is the pod, the hair pod, and the seeds can be 20 or even more in one pod. So these are the macromorphological features of Ruellia prostrata. So this is the outline. Daniel gave me this outline and asked me if I would add any other thing that was of interest. So from number 1 to 5 was this was from Daniel, but I added 6, 7, and 8 just to discuss a little more because I'm working on the medicinal plants. So my journey, process of patenting, I think you can read the outline. I don't want to go into that. So my journey. My journey started after completing my Bachelor of Pharmacy degree in 1979, then internship in 1990. And I would like to tell you that I was among the first 50 pharmacists to be trained in Kenya. So I was sponsored, I was seconded by the government, Ministry of Health. I was seconded to the Drug Analysis and Research Unit, School of Pharmacy, where I worked for 3 years. We were analyzing drugs for the government. If there were tenders, samples for tendering, we are the ones who did the analysis and gave the results to the government. And then checking, the government would check those which have passed and then now consider maybe cost. So we were at the scope, at the center of the drugs that were or purchased by the government. We are the ones who gave the reports of those drugs. So after working for 3 years, I got a government Commonwealth scholarship to pursue master's degree drug assay in the Department of Pharmacology at the All India Institute of Medical Sciences, New Delhi, where I did my master's from 1983-1985, and I worked on Roelia species. This was the species that my supervisor was working on. Actually, one student had done preliminary work, and I was the one that was the second student to work on this species. So I did much more of the pharmacology, but a first student just did the preliminary work, so I was the second student. So when I came back, I was posted to the Rift Valley Provincial Hospital. And by the way, if the government gives you a scholarship, even today, you have to work for 3 years. You have to give back to the government for 3 years. So that is what I did. I came back and I worked 1986, 87, and 88. And I got a challenge when I worked at the hospital. The challenge was most of the medicines were out of stock. There was not so much I would do. I would make an order. It would take a long time to go to Nairobi through the medical superintendent. By the time the drugs came, they were less than what I had ordered, and what was in stock was already gone. So I kept on telling our patients out of stock. And of course, I was very demoralized because as a pharmacist, patients would come, they would queue to see doctors, go to the lab. By the time they come for their medication, all we told them were out of stock. So I had to persevere for the 3 years. Of course, the job dissatisfaction. I was not very happy, and there's not much that I would do. So when I worked for the 3 years, I made up my mind. I said, I will not continue working in an environment like that, because what I wanted was to serve the patients. So if I would not serve the patients in a government hospital, I would have my own pharmacy where I would buy the medicines myself for my patients, counsel my patients, and interact with my patients with my ability to get the medicine. Getting the medicines was the problem. So I resigned after 9 years of work, started a community pharmacy called Friends Chemist, November 1988, and it is running till now. It was not, to be an entrepreneur is something else. Of course, we were very few pharmacists, so even to go to private practice, leave your salary in the government, not knowing where you are heading to, it was really a choice. But to me, it was not the money, it was job satisfaction. I just felt satisfied attending to the patients, helping them get good health. That is what gave me the satisfaction. So after 20 years private practice, where my children went to school, I got an opportunity to serve in academia. And this opportunity, actually, I did not even apply for it. Mount Kenya University wanted to start a Bachelor of Pharmacy program. It was the second university to start Bachelor of Pharmacy program after University of Nairobi. Nobody else was training Bachelor of Pharmacy students. So I was actually asked, I was requested to go and start that program. So I went into another field altogether after private practice, but there's a reason why I went to this academia. By the time I was joining Mount Kenya University, our profession, private profession business was already diluted. Quacks were having chemists all over the place, of course, chemists in quotes, and they were not doing the professional work. So I felt also again disappointed that, yes, we are now in this private practice, but quacks have come, they give like 5 capsules of amoxicillin, they didn't care. So again, I got dissatisfied with private practice. I said, can I be in a team that can train pharmacists so that one time we shall have enough pharmacists to flush out quacks? That is why I left my business. I did not leave my business even for any money, but I left the business because I had a call that one time, there must be a starting point. I had a call that one time we'll train enough pharmacists and flush out quacks. It has not happened, but it is going to happen because we have various universities now, government and private, training pharmacists. I think one day that dream will come to pass. So that is also again why I joined academia, to be in that team of people training pharmacists that we can have professionalism even in private practice. And of course, when you are in academia, research is part of academia. You will be relevant if you don't do any research. So after 2 years, that is 2009 and 2010, getting used to academia, It took me time because I was the coordinator, student admission, examination, Senate meeting. So I had again to get used to another lifestyle. So after 2 years, I said, this time I think I can make it. I can do some research. So anytime I got time, I went to the lab and started my research. So I started to work on this plant, because this plant that I worked on in India was promising. So what I decided, instead of just working on a foreign plant, why not go to the museum, National Museum of Kenya, find out if this Indian plant is in Kenya, because I got very good results in India. So my ambition or my passion was still on that Indian plant, which I felt that more work would be done. So when I went to the National Museum and found that this plant actually is found in Kenya, that was the start of my journey again. So I went, looked for it with the help of a plant taxonomist. I collected my first species in December 2010 and started the research. So after 2 years, 2010, 2011, almost, I think 2 years. After 2 years, I had very good preliminary findings, and the university actually sponsored me, paid my tickets, registration fee at the conference at the Johns Hopkins University, Maryland, USA, between 6th and 8th September 2012. So it was a scientific conference on medicinal plants and herbal products. So when I went to the US and presented these findings and met other scientists, I actually even got more passion to do the research. I said I will continue with this research because it actually exposes one to meet other scientists, researchers, and listen to what they do. So why did I do the research? The research is because there was a problem. And the problem is rheumatoid arthritis. As we all know, rheumatoid arthritis is an autoimmune disease. It's chronic, painful, leads to stiffness of joints, especially the small fingers and toes. But you see the fingers, if the fingers are stiff and painful, one cannot do any work. So the crippling and deformity of joints makes these patients be economically non-productive. And then, as we all know, there's no cure for rheumatoid arthritis. And there are a lot of adverse effects in the use when conventional drugs were used. So that was the problem. The problem that I was trying to solve. This is a condition. Can we have a medicine that is safe? That is all that I want. Can we have a medicine that is safe on these patients? Because there are some patients, when they are even given and arthritic drugs, they stop taking because the adverse effects of the drugs they actually have shared with me that are even worse than the condition itself. So my desire was, can we get a medicine that is safe, that these patients can take, they go about their normal activities as the rheumatoid arthritis is managed. So my justification for the study was to develop an effective and safe alternative. And natural products have remained the enormous reservoir of biologically active molecules. Actually, I looked at your website and you said that the past 30 years, no new medicines and microbial agents have been discovered. And I agree with you. Whereas the use of antibiotics or microbial agents has grown 40% in the last 1.5 decades. Actually, I agree with that statement because pharmaceutical companies used to finance research. But these days they don't. They have left research to universities and medical institutions, research institutions. They only come up when a molecule is almost getting a drug. They don't want to take all that money of theirs to spend on an unknown. So actually, natural products will continue to remain the enormous reservoir of biologically active molecules. Because you go to natural products, you will find new molecules which have never even been used conventionally. So why are they popular? They have minimum side effects, they are affordable, socially acceptable. So my justification was to investigate these medicinal plants, first for their safety and effectiveness, and as alternatives for the management of rheumatoid arthritis. So I will just share what was my broad objective in my study, but as I've said, for my PhD, I worked on 3 species, but I'm following up on 1 species. So for my PhD, I was to investigate the pharmacological and phytochemical profiles of extracts and bioactive compounds from Kenyan Ruellia species, and there were Ruellia prostrata, Ruellia bignoniflora, and Ruellia linearis So I had 4 specific objectives, which I'm not going into the details, but I had to do I did the chemical, chromatographic, and spectroscopic profiles. I determined antioxidant, analgesic, and anti-inflammatory activities. I determined in vivo toxicity, and I evaluated in vitro and in vivo and arthritic activities. So those are the objectives, and that is what I did for my PhD. So process of patenting. I'm following the items on the outline as given by Daniel. So the process of patenting is good to have family support. One of my sons, he lives in the US. He realized that I was so passionate with what I was doing. I told him this species had potential. And if it is in Kenya, I believe that if I work on it, I will get results. So he told me point blank, Mom, unless you protect your IP, you are going to waste a lot of time. You start off by protecting your IP. He told me if I was very sure that there was something that would come out of this research, then we start the protection of the IP, intellectual property. So actually he started for me the patent process in February 2011. We used advocates, we used Simba and Simba advocates to start the process of patent application. And then currently we are using Kibunge and company advocates. It's good to have these advocates because somebody can come up and pick your research idea. It's always good to have some advocates so that in case something like that happens, they can protect us. So we use Simba and Simba, but currently we use Kibunge. So before any process of patenting, you go to the website of KIPI, that is Kenya Industrial Property Institute. You go to the website. The website has so many forms depending on what you want. So the form for patent is called IP3. So you go online form, KIPI website, IP3, you get the form for patent. So it has also many things to do. You just fill as per the requirements. If they said put the abstract, put the rules are there, the guidelines are there as to how to fill these forms. So it took some time, and we managed to fill the form. We managed to put our idea because they want something new. What we are putting up as it should be new. It should be your real thing. Not copying somebody else's. So after that, we published, the abstract was accepted and it was published in African countries, and it took 6 months for the results to come back saying that nobody is working on that, on this particular species. So that is how the IP3 form looks like. You go to a website, it is the Industrial Property Act 2001, and the first one is a request for a grant of a patent. This one also is used for request of registration of a utility model. So you tick what you want, and the form is self-explanatory. Just fill the form, whatever is written in that form, you complete it. So when you filled this, the results came back after 6 months that nobody was working on this species. I got a shock of my life when a Filipino researcher came up and said that I was working on his species. And he was working on Ruellia tuberosa. It was a difficult moment for us. That is why I say it is good to use lawyers. So I had to sit down and now challenge the challenge. I had to put all the facts from the time I was a student, what I had accomplished, even the conference in the US, I had all the evidence of whatever I had done. And of course, I had to defend myself and say, my species is Ruellia prostrata, and the Filipino researcher's species was Ruellia tuberosa. Even took the pictures of the two species. It was something that of course, it was not interesting. It took another 6 months to challenge the Filipino challenge. But finally, the Filipino kept quiet because I had enough facts to tell him that my species was not his species. So when we now got the green light to continue with the patent process, I had all the hope. All along, I've had the hope that one time a medicine will come out of this species. So when this medicine comes out, who is going to manufacture it? So we founded another company called Jomo Pharmaceutical Limited, that is the website, which started processing the patent matters. Because initially our son just started paying for the patent matters, but when we incorporated this company, it is the company that we are trusting that is the one that finally will manufacture the medicine that will come out of this species. So I'm one of the founders of this called Jomo Pharmaceutical. You can go to the website, you will see interesting things that we do. So finally, after starting the application in February 2011, finally the patent was granted in August 2016. So you have just to be patient. It takes a long time, but one has to be patient. So the patent, we are not patenting that this plant is active. We are patenting that we are making a formation of a pharmaceutical composition for arthritis, inflammation, and pain. We cannot patent a plant that was made by God naturally. So we patented a formation of a pharmaceutical composition. It is a long abstract. It's a long abstract. So, but the main thing that a formation of a pharmaceutical composition for arthritis, inflammation, and pain. So these are the legal fees. You go to KIPI, you'd find the general legal fees for local people, foreigners. All these things are on the website, and I don't want to go into the details. So I just brought these slides that you can see what is the fee for what is the fee for what, and So as I continued with the research, and being at the university, as I said, the research needs to, the academia goes together with the research. And in the end, any of you wanting to pursue being lecturers, just remember that PhD is mandatory. So I did my research. I tried to, I had passion to pursue my PhD studies as I continued with my research, but unfortunately at MKU, being that time I'd risen to the head of Department of Pharmacology, so administrative duties and many other things, it was not possible for me to actually register. I tried to register, but I reasoned with myself that registering with all these responsibilities, it was not going to be possible. But I would run to the lab, just do a few things, but I didn't register as a student. But when I realized that time was going, I was looking forward to registration as a student. So when JQuat advertised in May 2013 that there were slots for PhD studies, I applied to be a private student. I was ready to pay for myself. So I applied to pursue the PhD studies in JQuat as a private student. So actually I started my studies May 2013, went back to class for one semester, being taught the cuts and everything. So I did that one semester and passed my exams, and I was to start the research. But God was on my side. Immediately I paid the fees, the May-August semester. I was again hired by JQuat as a senior lecturer in July 2013. And I got full scholarship to pursue my PhD studies. So I paid fees only for one semester. So my research has been having God's blessings. So I got full scholarship. The JQuat paid all the fees and gave me 200,000 per year for 3 years for the research purposes. So as I continued with my studies, Chebu, I'll call it Chebu Pharmaceutical, but I'll call it Chebu. Chebu funded a 3-month course on continuous professional training in drug discovery and design at the University of Washington, USA. So I went there to find out when we are getting drug discovery, what are some of the things. So I learned a lot from the University of Washington for those 3 months. So again, God again was on my side. The university advertised for research grant for staff. I wrote a proposal that was successful, and I was awarded a grant of 4,230,000, running from 2016 to 2018. JICA, that is Japanese International Cooperation Agency, also advertised for grants. In 2018, again, I wrote a proposal that was successful, and I got a grant of 860,000. So these funds actually helped me accelerate my research studies, because at that time I began working on animal experiments. Each albino rat is about 400 shillings, whereas a mouse can be 300. So it was going to be very expensive without research grant. So the research grant that I got from both JQuat Staff Research and JICA accelerated my research studies. So I didn't have issues of money. I just planned my work, knowing that the money was there and it was going to help me move forward. So the process of screening, before one does any research in this particular area, one must know the problem statement. What is the problem that you are trying to solve? It must be very clear. Don't just pick on something. Is it worth studying? How does this problem affect the people in Kenya, that is locally? How does this problem affect Africa regionally? How does this problem affect the worldwide population? That is, so what is the prevalence of this problem? Is it, for example, if you're just going to work on a problem that is affecting a small region in Kenya, maybe some part at the coast, is it worth studying? Yes, it may be worth studying, but really if you're going to have a problem that is going to affect people locally in Africa and the whole world, then it is really a problem that you need to solve. So that is also one thing. How does this problem affect people, local people, regionally and worldwide? And for the problem statement, once you know what the problem is and you are and one is convinced that it's a problem that needs a solution, you must have the solutions to the problem. Nobody's going to give you the solutions. You sit down and say, if this is the problem, I'm the one to have the solutions to this problem. So how do I go about it? Therefore, you must have a justification to carry out your study. Without a justification, you are wasting your time. So we are looking for new medicines. I've already said that new medicines are coming from natural products. They're the ones that are producing new compounds, new molecules that can be used in drug development. The source of information as far as natural products is concerned is normally folkloric. So you'll find that herbalists are saying they use this medicine for this particular condition. The only advantage of the natural products is that if they have been in use for a long time, then the safety may be guaranteed. I'm not saying must be guaranteed, it may be guaranteed. Because if patients have used this medicine for a long time, it means some safety, there's some amount of safety that can be guaranteed. So you are starting off with some safety, then starting off with a compound. If you start off with a compound, we have no idea how safe it is. So there will be rigorous experiments to be done. But when we start with the medicinal plants, these are natural. We start off with some safety aspect that if patients have used these medicinal plants for a long time, then there's some safety that can be guaranteed, because safety is a very important thing. When we're trying to look for a new drug, safety is number one before even efficacy. Safety is number one. So once you know you are working on a particular plant, I will talk about plants because that is my area. You must know the botanical name of that plant. There's no way you're just going to look at two plants and say these two plants are the same. So you need a plant taxonomist to identify the species before working on it, because some species may look as similar and yet they are not the same. So you need a plant taxonomist to identify the species before working on it. So after identifying this plant, and you know, maybe folkloric medicine says this drug is used for anti-diabetes, as an anti-diabetic or whatever, you must do literature search on whether such a plant has been studied, or as this plant has any other activity that was reported, but was mentioned in folkloric medicine, but has not been worked on. So the literature will tell you this plant, whether it has been studied, even that there are some reports regarding the activity you are looking for, or you may even find that it is reported to have other activities in folkloric medicine which have not even been worked for. So literature search is a lot. You have to read. And I thank God for you, the current generation. You have Google at your disposal. Google anything, it will pop up. Thank God for technology. During our days, we physically went to the library for literature research. We also had typewriters. There was nothing like delete, cut, paste, save, and all those things. You made a mistake, you started afresh. There was nothing like what you have these days. So we thank God for technology, but during our days, you can imagine if you wrote a proposal or did your thesis, it was really work. So from literature, you can also, as I've said, you can also check any other activity, the activity you are looking for, or you can also check any other activity that has been mentioned and has not been studied. We normally say the gaps between the research. Is there something that has been mentioned but not studied? So you can also check that. So for Kenya, consult the National Museum of Kenya. It will tell you the records there will tell you the local distribution of the plant species. That is exactly what I did. There was a description where this plant species was. I explained to the plant taxonomies, and we actually went to those areas searching. We searched in many places and then we found that particular species in that area because they are voucher specimens. The voucher specimens are normally deposited in the herbarium or the National Museum. So when you get your plant, after identification by the taxonomies, you must always keep a voucher specimen in a herbarium, because it is this voucher specimen at the National Museum of Kenya that made me know that this plant can be found somewhere else. So voucher specimens must be done and deposited in various, in a herbarium, wherever it is, can be a JKUAT, University of Nairobi, National Museum, wherever there's a herbarium, you always keep a voucher specimen. So once the plant is obtained, it's normally we dry under a shade. We don't like direct sun because it may lead to deterioration. Grind the plant material to cause powder and do phytochemical analysis. Phytochemical analysis is important because there are changes in plant composition. The compounds in the different plants will depend on soil composition, climatic changes, geographical location, genetic makeup of the species. You may find that when the species are in two different geographical locations, you may find that the composition, the quantity of the active compounds may differ. So it is always good to do preliminary phytochemical analysis. And we do start off with the extraction. We start off with the sequential extraction, where we start off with the nonpolar solvent. So you get one quantity, extract with N-hexane, it will remove all the nonpolar compounds or petroleum either. Then you increase the polarity like dichloromethane, then methanol, which is very polar. That is by cold maceration. That is without heating. Aqueous extraction is normally done with some kind of heating because cold water normally does not extract. So what is the use of the extraction? Is to find percentage yield. Percentage yield will tell you that this is the amount, these are the compounds that are in bigger amounts. Because if you are going to have a percentage yield of 0.2%, how will you do your experiments? How will you do your in vitro, in vivo experiments? So if you have a yield like 10%, 8%, 20%, then it means that this particular plant has more of those compounds which have given the highest yield. And then these are the ones that will be good for one to continue with the other experiments, both in vivo and in vitro. So before any medicine is used in humans, it must go through animal experiments. These are called preclinical studies. And there's something we call the 3 R's in animal research. So before even somebody touches any animal, the 3 R's must be taken care of. One is replacement, reduction, and refinement. Replacement methods means you avoid the use of animals in research. Are there other protocols that are going to test your activity to give you preliminary findings before you go to the animals? There's no need sacrificing animals when we have not used other methods. For example, let me just say antimicrobial agents, now that you are people of antimicrobial agents. There's no need of testing on a human being when you have not tested in the lab, using the disc, whatever method that are used in the lab with the reference drugs to find out how they act. So those dishes, you know what I'm talking about. So there's no need of going direct to the animals when you have not tried even the one in the lab, using the methods in the lab in microbiology. So we have what is called a replacement. Methods which avoid or replace the use of animals in research. We start there first. If activity is there, then we move to the animals. The other one is called a reduction. The use of methods that enable researchers to obtain comparable levels of information from fewer animals. Yeah, is there a protocol that will use fewer animals to give a hint of the activity? And then we have refinement. Methods that elevate or minimize potential pain. There are rules about animal experiments. We don't cause animals pain for nothing. Don't make them suffer for nothing. We don't distress them for nothing. So are there methods that can elevate that? So that we use those methods first, then when the activity there's a potential, so now we can move on to the animals. Normally we select one or two animal species. Most of the times we use albino mice or albino rats. They're the ones that are mostly used. They are small in size. Therefore, even if you have a small amount of extract or a drug, you'll be able to give to these animals and they'll give us some results. And actually, the mice have the nearest genetic makeup to humans. If you did not know, you can go and Google. Mice have the nearest genetic makeup compared to the human. So a drug that is going to kill a mouse, it is most likely going to have adverse effects on a human being. So before using these animals in preclinical studies, you must seek for ethical clearance. Must be cleared to touch the animals. You don't touch any mouse or rat without ethical clearance. And in Kenya, we can use KEMRI, University of Nairobi, KNH. So that approval must be done. For my case, the approval took 9 months. There's nothing you can do, everything. You just have to be patient because they have their own calendar when they meet, when they review, when they look at the reviewed proposals. There's nothing you can do about it. You just have to wait. Mine took 9 months. So, before handling animals, one must know how to administer the drugs, how to euthanize the drugs, the animals, and that can be done by registering for a city online test. That is collaborative institutional training initiative online. There are online modules which will tell you how to handle any animal, from a rat to any rabbit, just any animal. You will be told if it is a rat, if it's a rabbit, this is how much volume to give. If you want to draw blood, this is how much. Just handling of the animals. You can register for those city online test modules where you will be taught how to handle the animals. So, as you are waiting for the ethical clearance, as I've said, it can take even 1 year or whatever time. You design some in vitro tests that you do not waste time. You can do the preclinical phytochemical screening. You can develop these other tests, which I said, which you can do before using the animals. So in between, you can be doing something. So when the ethical clearance comes, you'll also have gotten results from this in vitro test. You'll be able now to move on with your protocol. And every drug that is used in humans has passed through animals at one stage or another. So whatever activity you are looking for, whatever your problem statement, look for protocols, animal models that have been designed to detect that activity. And that is what you will use. So once the activity is confirmed and you've gotten clearance, in vivo models can be tried after ethical clearance. So for my case, I'll just share from my journey to do the antioxidant activity, because we know drugs or compounds or plants that have antioxidant activity have been reported also to have anti-inflammatory activity. So before going straight to the anti-inflammatory activity in the animals, I had to do the in vitro test. What is this in vitro protocol that can detect antioxidant activity before I go to the animals? So we have something called DPPH, is a long name, scavenging activity. It is a compound. It's a compound that has a nitrogen-centered free radical, which is stable. So it will scavenge radicals from the antioxidants. So when it is purple, when you give your antioxidant, it will scavenge the H and make a stable covalent bond. And there is a color change from purple to yellow, and the reading at 517 nanometers will determine the amount, the scavenging activity, where one can calculate the antioxidant activity. So that is one in vitro test that I did to find out if this compound plants had antioxidant activity, and they had very close to ascorbic acid. So for arthritis and arthritic activity, there's also another in vitro test. This is what I said, you look for other protocols that you can use other than animal experiments. So for arthritic activity, there's inhibition of protein, denaturation by heat. You can either use bovine serum albumin or egg albumin. I chose bovine serum albumin because this standardized. A problem with egg albumin, it may depend on what diet the hens are given, so it may not be very standard. So I prefer to use a standardized protein, which is bovine serum albumin. So it was reported that if a drug has an arthritic activity and you put protein and you heat that protein, that drug will inhibit the denaturation of that protein by in some ways a protocol that has been reported. So the turbidity is recorded at 660 nanometers. So process of regulation, as I've said, I've done preclinical, but I'll just share some clinical trials, review and approval. For my research, I am between preclinical and clinical. So there are so many processes between preclinical and clinical. That is where I am, because you have to have the drug approved by the Pharmacy and Poisons Board anyway. So I'm in this process. If the Pharmacy and Poisons Board approves my drug, then I can write a proposal for clinical trials, which will go under review and approval. So I said that we must do preliminary phytochemistry to find out the nature of compounds in these plants. Plants have compounds like alkaloids, saponins, terpenoids, flavonoids, phenols, tannins, steroids, and many others. Why do we look for them? We look for them because this study guides one on what solvent would be used to reconstitute the extract for animal experiments. If the compounds are polar, then we use a phosphate buffered saline pH 7.4. If the extract that are in large quantities are organic, then we may use phosphate PBS, phosphate buffered saline, with 1 or 2 drops of the dimethyl sulfoxide. So it will give us that hint, because once we have this compound, they must be reconstituted in some solvent to be used in animal experiments. So once we know the nature of these compounds, we'll know whether we shall either dissolve them in phosphate buffered saline, or if they are organic extract, we shall use some 1 or 2 drops of dimethyl sulfoxide to help them dissolve in the aqueous solvent. So generally, this is what happened, or this is what can happen when we are dealing with plant material. Sample preparation, of course, after collecting, drying, and grinding, there's extraction. As I said, the extraction can either be of the organic solvents or aqueous or distilled water. So once extracts are obtained, there are many things that can be done with this extract. One can be chemical analysis, where we're going to do the qualitative analysis to find out which type of compounds there are. And then we have quantitative analysis to find out the percentage yield of these compounds. These crude extracts can also be subjected to chromatographic analysis, where we're going to separate the type of compounds in these extracts and get what are called fractions. So these fractions can be subjected either to bioassay or can be subjected to spectroscopic analysis. The extracts can also be subjected to spectroscopic analysis and bioassay. So once you get the extract, there are very many things that you can do. Initially, it is always good to do the chemical. As I've said, it will give you the nature of the compounds. It will give you the percentage yield to know which of these compounds are the one that you are going to deal with other tests. That is generally what one can do. So once the activity in the in vitro test has been established, before going to animal experiments, we must test the toxicity, because it is better to have a drug that is less effective and safe than to have a drug that is effective but very toxic. So toxicity studies are carried out before subjecting the animals to any studies. So there are acute toxicity studies, which are observational. To give one dose is given The animals are observed for 4 hours, especially with the emphasis of the 30 minutes. So the one dose is given to the animals, usually female albino rats. So once one dose is given, the animals are observed for 4 hours, and especially the first 30 minutes, to see any actions like lacrimation, defecation, convulsions, coma, the fur rising on the body. There are so many things that you can. The animals normally, when they are in a cage, they like playing together. When you find one animal just alone in a corner, all those things, the animals are observed for 4 hours, and especially the first 30 minutes. That is why we say acute toxicity studies. Then after that, the animals are left alone. After 24 hours, come and look at the animals again, find any death, find any adverse effects, clinical symptoms. And then again, these animals are observed daily for 14 days. So if the medicine has no effect throughout after the 30 minutes, 4 hours, 24 hours, and 14 days, then we can say the dose is good enough for this is safe in this acute toxicity study. Normally start with 300 milligrams per kilogram. That is the current protocol. If there's no adverse effect, then 2,000 milligrams per kilogram is tested. And if the animals are safe with the 2,000 milligrams per kilogram, we can say this plant does not have adverse effects in acute toxicity studies. Then we have what is called subacute toxicity studies, where one dose is given to the animals for 28 days. That is where work is. Observation every day, weigh the animals every day for 28 days. After 28 days, the animals are sacrificed using carbon dioxide euthanasia, and then blood is drawn quickly by cardiac puncture for hematology and biochemistry. Then their internal organs are removed, the heart, the liver, kidney, the spleen, intestine, sex organs, all the vital internal organs are removed. There's what is called gross necropsy, just to look at them. Do you have any tumors? Do you have any paleness? Just observation, just the way we do post-mortem. So after that, the internal organs are preserved in formalin, and histopathology is done later on, where thin slices are made and observed under a microscope. The relative organ weight, body weight are also observed. So for in vivo tests, I said I'm not going into the details. For in vivo analgesic studies, there's what is called tail flick method for acute pain in mice. There's what is called formalin test for progressive pain in mice. So those are the two models for in vivo analgesic activity. Then in vitro anti-inflammatory activity, there's what is called carrageenan. It's a substance that is injected into the foot hind paw, and then there is swelling, and by use of what is called a digital plethysmometer, one can be able to determine the swelling by displacement of water. For the acute pain, an equipment called digital analgesiometer is the one that will detect when the animals flick the tail. So for in vivo and arthritic activity, there's something called a complete fruits adjuvant, which is when injected into the rats at the base of the tail, it will, after about 10 days, it will show all the arthritis conditions, like the ones that are seen in human. This particular protocol is the nearest protocol that gives the likeness of rheumatoid arthritis in patients. So hind paw swelling, hide joint thickness, grip strength, arthritis score, body weight, non-articular symptoms like nodules, just the way it happens in humans. All this can be assessed. And of course, comparing with the untreated control and comparing with the standard drug. For arthritis, I used methotrexate as the reference drug. For pain, I used acetaminophen, ibuprofen, and diclofenac as the reference drugs, and morphine, because I used both opioid and non-opioid analgesics. So data analysis, you always say what your statistics are. Normally it is mean plus standard error of mean, and you always have a value, a p-value, where you'll say that a p-value of less or equals 0.05 will have significant difference. So you don't do your research without analysis, and you must have a p-value where you say, if this value is this, then there will be a difference in the activity. So for chemical, chromatographical, and spectroscopic, TLC, I think you are familiar with the TLC. It's the one that separates the compounds, and normally there is normally incorporated with the fluorescent indicators. So first of all, when you're running the TLC silica gel, the commercial TLC plates are coated with normal silica gel. So after separating the compounds using different solvent systems, they are viewed under visible light. Some compounds are colored under visible light, or they can be observed under UV 254 nanometers, 365 to see any fluorescence. And if there is no fluorescence, we can derivatize the compounds by using spraying reagents like vanillin iodine. So various solvent systems were used. I also don't want to go into the details, but this can help. There are so many solvent systems. There is no literature which will tell you use this. Everything is trial and error. It's trial and error all the time. So these are the solvent systems that worked for my separation after trying very many of them. So the spraying reagents, as I said, vanillin, concentrated sulfuric acid, or iodine, they can be used for visualizing the compounds because they derivatize the compounds, and now they can be visualized in the UV. So there's one thing called RF value. If compounds are done and if tests are done under same conditions, compounds which have same RF values are normally the same compounds. So get the distance traveled by the compound. This is distance traveled by solvent front. It gives you the RF value. So it tells particular compounds have different RF values. Then spectroscopic profiling, I use the FTIR, Fourier transform infrared profiling, to determine the functional groups. What are the functional groups in this? So the region between 35 and 1450 is called functional group region, and most common vibrations are here, like CH, CO, C double bond O, COOH, all these are found in between 3500 and 1450. But from 1500 to 800, we call it the fingerprint region, where there are most complex vibrations. And actually, this particular area is the one that distinguishes compounds from each other. So that was also done. Then for spectroscopy, GCMS is also used to determine the compounds compared to the National Institute of Standards library. We have compounds in the library, so when a GCMS analysis is done, the compounds will come out and will be compared to the ones that are in the library. So what are some of the things? Retention time, molecular weight, molecular formula. We also have something called similarity index. So this is the one that's going to determine what are the compounds that are found in this. The only problem with the GCMS is that we use high temperatures, around 250 degrees centigrade. So we may have compounds or broken parts of the compound. That is the only problem of GCMS. Some compounds which are heat stable will pass and be detected, but some compounds that are heat labile will be broken by heat, so we'll end up getting metabolites. So it is a good way for spectroscopy, but it's not give you all the time the actual compound because of that heat. That's under high temperatures. So once this once these drugs were found activity, found the nature of compounds, then there's application for a trademark. Application for a trademark is also obtained at KIPI. Also the same forms, they scored from TM2. The forms on KIPI website are self-explanatory. You just fill the forms the way they are, after depending on what you are looking for, and then you have application for registration of a trademark. They tell you the amount of money for locals and for people outside, then the fees, and then everything is on the website. I'm just sharing with you that if you go to KIPI, whatever you are looking for is there, plus the fees. So once you get your trademark, you will find some drugs have R or TM. So R means registered trademark and TM just means trademark. So when I did my application, in 2019, at the beginning of 2019, and by December, I got a trademark called Jomo Tip. It was very hard to get a name. because I wanted a name which starts with Arthro something, Arthro Care. Every name which starts with Arthro was already booked by some people. So when I got frustrated, and of course, when you are doing that time you are paying money. We were just with a friend of mine. After getting frustrated, we had come from the Sharia place to find out this trademark. Then we just discussed in the car as we were coming back to JQuad. What is arthritis in Kiswahili? It's Jongo. Why don't we, nobody will know what Jongo is. So we just did Jongo tip and the name went straight away. So you have to think also very fast. So this is Jongo tip, Kutib Jongo, treatment of joints. That is how my name came about, Jongo tip. So you can either be Jongo tip registered trademark or Jongo tip TM. So I'm just trying to tell you the signs that you will find when you have a drug that has R or TM. It means it's a trademark and it's a trademark sign. So after that, now this is where I am. I'm on PPB dossier application. There are also forms. Everywhere there are forms and forms and forms. This is the dossier application form with the PPB, because PPB must approve this complementary medicine for use in arthritis. It has many things. I just give the particulars of the applicant, particulars of the product, particulars of the manufacturer. That is why we made Jomo Pharmaceutical, because they will need particulars of the manufacturer, composition of the product, quality control of the finished product, stability studies, pharmacological and toxicological information. So all these things are needed by PPB. So actually this is where I am now. I've already submitted my dossier and it's very expensive. It's 53,000 to have your dossier examined, and I'm hoping that one of these days I will get some good news from them. So clinical trials, I've already told you, clinical trials I've not done because I have to get that approval from the PPB, but just generally this is what happened. This I got it from FDA online. So what we do is preclinicals, where we have in vitro, in vivo, using the animals, that is the preclinical. Then once there's activity and there is safety, there are two things, there's safety and there's activity, we can go to phase 1 clinical trials where we use 20 to about 100 participants. Normally this one is, most of the time phase 1 are normally healthy volunteers. So these healthy volunteers go for their blood analysis, liver function test, kidney function, heart function. They are healthy, all these parameters are taken, and then they are given one dose for 1 month. So after 2 weeks, they can go for a checkup, and then after 1 month, just to find out what is this drug doing in a healthy volunteer. So once there's no effect, so it is actually safety now. Once there's no, because here the safety we did in animals, but now we are going to patients. Once these healthy volunteers get this medicine and there's no effect on their normal parameters, then it can go to phase 2. Phase 2 is normally 100 to 1,000 participants. Most of the time, checking the kind of condition that the patient has, if it is hypertension, arthritis, we want to find out the effect of the drug in now a patient. And phase 3, we can, more people, we can also have a patient may have arthritis and hypertension. So drug-drug interaction. So we want to see drug-drug interaction, we want to see drug-disease interaction. Then FDA approval after more than 3,000 participants, it can be submitted for FDA approval. There is always post-surveillance after the drug has been confirmed. There's always also post-approval surveillance, because you can find after a drug has been approved, you'll find that some other side effects can come much, much later. And the drug can actually be approved and the approval can be disqualified. So there's this post-approval after experience. So this also just from the website. These are the first one, early discovery going on, the lab, the animals, phase 1. As I said, safety and best dosage levels are determined. Then response to new treatment is recorded and analyzed. So now in this case, we can use placebos, we can use standard drugs, we can have a group with the placebo, we can have a group with a drug that is already in the market, we can have now your drug being tested. So, and then as I said, post-approval treatment is marketed. There's also post-approval. Any new drug that has gone to the market, it must be checked. If any signs come from different corners, then it may be, those signs may be related to this particular drug. And as I said, if the signs are too much, the drug may. So this one, I don't want to go into the details. It is actually giving more of phase 1, determine the metabolic and pharmacological actions. What are the data focus are vital signs, as I said, check the blood, the liver function test, the whatever, up to 1 month. So phase 2, several months, phase 3, several years, and so forth. And phase 4, ongoing following FDA approval. So it will take a long time for a drug to be considered safe. So what is the ongoing work? The ongoing work, I'm trying to get the compounds, the actual compounds in Rheumatoid prostrata using NMR analysis. So far, we have managed to identify beta-stigmasterol, which is one of the steroids in many plant species and many Rheumatoid species. Using NMR analysis, we have gotten beta-stigmasterol, but now we have approximately 10 compounds which have been separated using column chromatography. And what is happening, we are going to do, NMR has so many, it's not just proton NMR and carbon NMR. We have also COSY NMR, HCQC NMR, HMBBC NMR. So NMR analysis from all these, we put the peaks together, we put the information together, and an expert, I have a chemist, that is now not my area, an expert will be able to take all the results from proton NMR, carbon NMR, COSY 2-dimension NMR, to put together to come with the particular structure. So far we have gotten beta-stigmasterol, and we are looking for compounds. We are hoping that either we can have a novel compound or a compound of non-compound, activity of non-compound not reported. Yes, I may have a compound which has been reported in the chemistry library, but has not been reported to have anti-arthritic activity. So I'm really having hope that of these 10 compounds, something will come out. Some compounds will come out, known or unknown, but if known, the activity of arthritis has not been reported. So still that will be something worthy. So we have what is called in silico studies. I put this here because in silico studies, this is what I learned when I went to the University of Washington. It is one of the studies that I learned from there. We are all in the computer, in the technology. We are all in technology world. So in silico studies, just means that we are using network pharmacology approaches. We are using the computer to work for us things very fast. Can test thousands of compounds in 1 minute. So what happens? There are software, for using in silico studies, which are going to investigate the targets. Like compounds, as I've said, library, chemistry library as compounds, which you can use GCMS to find out what they are. But also we have libraries of targets or receptors. So for arthritis, what are the targets or what are the receptors that have been documented that a drug that is working on this target or receptor could have an anti-arthritic activity? So using computer simulations or network pharmacology, you are going to get, as long as you have the compounds and the structure of the compounds, they are going to match these compounds with these targets that have been documented. And the one that will give the best fit is the one that can be studied further in both in vitro and in vivo. So actually people and researchers are using these methods. They will reduce the number of animals that you are going to try, trial and error. You will work in a way that you know very well this compound is sitting on this target, and this target, this is the animal model for this target. So it is a new way of getting information. So there are putative targets, as I've said, which are in the libraries. Meta-drug is a way that just go and Google meta-drug. It will be able to tell what are the putative targets that are there for various diseases, whatever disease you are looking for. So once you get the compounds that are going to target these particular receptors, For this particular case, it was rheumatoid arthritis. So there are features, we call topological features in these studies that have different things like degree, node betweenness, closeness, K-corness. There are things with this software that will tell you the best, the one that is giving the best fit for that particular receptor. Then we have the binding efficiencies, and molecular docking is one. It's one that so when we do when molecular docking, I'm going to talk about the molecular docking simulation. We have what is called Lipinski's rule of 5. You can also Google it. It is a rule that decides that is this drug likeness or drugability? Is this compound of yours, can it be a drug? It will determine the chemical compound, pharmacological, biological, and find out if there are chemical properties or physical properties that would make this drug orally active in humans. This is all about in silico studies. So using in silico studies, you'll be able to find out if this compound can actually be a drug in humans. So it will describe the molecular properties, it will describe the pharmacokinetics, which is absorption, distribution, metabolism, and excretion. The only problem with the silico studies, it will not tell you if the compound is pharmacologically active. It will tell you this compound fits properly on this receptor. The absorption is okay, distribution is okay, metabolism, because even there are enzymes, the cytochrome P450 enzymes targets. So it will tell you everything else apart from pharmacologically active. But the goodness that if you are going to find a compound that is safe, absorbed well, distributed well, metabolized well, then now you can go to the lab to actually test the activity. So it will have reduced so many things for us. So in silico studies, we are being impressed in drug discovery to give you a compound that can fit properly on a receptor and has best pharmacokinetic properties. So now you can go ahead to go find out the pharmacodynamics. So this molecular docking, I did various receptors using the library, but I'm only going to discuss prostaglandin synthase 2 as one of them, just for you to appreciate what pharmacokinetics, what in silico studies can do. So these are my findings on prostaglandin synthase. I used compounds from GCMS. I used compounds from GCMS to do molecular docking, but the compounds that really have a lot of sense, if you want funding for research, the compound that will come from NMR. If these compounds of mine, which came from GCMS, if they had come from NMR, I would have gotten a National Institute of Health NIH grant from the US. They were willing to give me money to develop this drug if only those compounds were NMR. They said the research was good. I wrote a proposal which was accepted, which I was told was innovative, but they just told me that the reviewers said, if only those compounds were from NMR, I was going to get a lot of money for my research. But okay, there's no problem. In research, you don't give up. I had now to go back to the drawing board and start the chromatographic separation. And now I'm on compound elucidation. So this is what happened. This is one of the receptors. The receptors, the prostaglandin, that is cyclooxygenase enzyme 2, which is one of the enzymes that is used as an anti-inflammatory, where anti-inflammatory drugs sit. So there's a 3-dimensional interaction. One of the compounds from the GCMS was hexadecanoic acid. I'll just say hexadecanoic acid propyl ester. It's a long name. So this is C. Then I have hydroxychloroquine. That's a drug used in rheumatoid arthritis, methotrexate, and sulfasalazine. Methotrexate is the first-line drug in rheumatoid arthritis. So I have one compound, and I'm going to compare with the 3 conventional drugs used in rheumatoid arthritis. So this is what happens. That is what in silico simulations do. They are very interesting results. They'll tell you how the drug fits in the receptor in 3 dimensions. This is the hexadecanoic acid propyl ester. This is hydroxychloroquine. This is methotrexate. This is sulfasalazine. So you can see how you can actually see in 3 dimensions using in silico simulations how this drug is sitting in that receptor. That is what in silico studies do. They tell you how it fits in that receptor. So I can say this is hydroxychloroquine is sitting on the target of prostacox. Let me say COX-2. COX-2. Hydroxychloroquine is sitting there, sulfasalazine, methotrexate, and this compound coming from Rheumatoid prostrata also sat in that on that target. So it has some activities showing me that it can act as an anti-arthritic drug, because, let me say in this particular case, as an inflammatory drug, because it was also able to sit in that pocket. You can also have two-dimension interaction. This is the compound. This is just another naming in chemistry, but it's the hexadecanoic acid propyl ester. So now the two-dimension interaction, this is what in silico studies do. They'll also give you the two-dimension. The advantage of two-dimension, every receptor, the pocket, the receptor has linings of amino acids in the receptor. So these are the pockets. Like here we have histidine at position number 214, lysine position number 211, histidine 207. In the two-dimension interaction using the computer simulations, we'll be able to tell you that the pocket of COX-2 has this kind of amino acids in the pocket. So this is how this compound that came from my plant sat on that pocket. So it can it's still telling us that it can sit on COX-2 target. Come to hydroxychloroquine. These are the type of amino acids in the pocket. So of what use is this to us? This is now what happens. We can check the hydrogen bonding of these compounds in these pockets. So hydroxychloroquine, apart from other amino acids lining the active site, we have leucine at 352, adenine 527, valine. You can see that when we come to our compound, valine, all that are colored yellow. Leucine 352 is here. Adenine 527 is here. Leucine 359 is there. So using computer simulations, you can be able to tell that your compounds are sitting in that target, and these are the kind of amino acids lining the receptor, and you can compare your compound with the standard drugs. So it can tell you that this compound that you have got has some anti-inflammatory activity. So this is also another presentation using in silico studies. We have talked, it's going to tell us about lipophilicity potential. So the areas that are purple and blue means this compound is lipophilic. It's mainly lipophilic. So wherever it is going to sit in the target, it will sit in the targets where we have amino acids that are lipophilic. So this is the compound. This is hydroxychloroquine. This is methotrexate. We know methotrexate is polar. So you can see methotrexate has orange and red. So it is more of hydrophilic. What I'm just trying to share with you is using in silico studies, you can learn a lot from your compounds even before you go to the lab. What is the nature of these compounds? What are the type of bonding in the active site? How are they fitting in the receptor using three-dimension molecular docking? Two-dimension will tell you that kind of amino acids. And then lipophilicity potential will tell you if your molecule polar, is it non-polar? So all of this you get from in silico studies. So the target validation, once you get the in silico studies, as I've said, we can only do up to pharmacokinetics. We can do how the drug sits on the receptors. We can do how the drug sits on the cytochrome P450 enzymes. We'll be able to find out which are the cytochrome P450 enzymes are metabolizing the drug. When we find this in the literature, people did all these things that we are doing. So once that is done, I said we are not going to, it will not tell us if the drug is active. It will tell us that the drug has the proper pharmacokinetics to be developed into a drug. But now for the drug, work has to be done. So for in this case, we are looking about anti-inflammatory activity. We want to find out what are the effects in vitro tests on inflammatory mediators. And this is this plant working on cytokines, tinea necrosis factor, interleukin IB, IL-6, IL-10, IL-17. Is it working through the COX pathway like the one I've just shown you, prostaglandin E2, leukotrienes? Is it working on the immune system like interferon gamma? There are some blood parameters in rheumatoid arthritis patients. Before rheumatoid arthritis patients are put on drugs, these parameters are normally taken: C-reactive protein, rheumatoid factor, anti-CCP, erythrocyte sedimentation rate. They are normally determined in patients before the drug is given. And then as patients take the drug, they are occasionally determined to see whether the drug had an effect on these parameters. So this can be done in rats, as we have said. We cannot go straight to the animals. This can also be done where this will be tested in rats and find the effect on these parameters. So once mechanism of action is established, we need permission to carry out preclinical trials in a non-human primate. So developing a drug is a very expensive, time-consuming, needs resilience. Like, I think I've used over 10 million for the time being, and I've done this study for more than 10 years, and yet I've not even gotten to the human primate, but I'm still hoping that before my patent expires, this will be done. Actually, the bottleneck for the time being is the PPB. If the PPB can give me an okay, I'll develop a protocol for the clinical trials. So this work, as you can see, it is not work for one person. I was funded. I was funded by Jomo Kenyatta University of Agriculture and Technology. I was funded by JICA. I was also funded by my company, Jomo Pharmaceutical Limited. This is where I got money to do the research that I've done. And as I've said, so far, we have used more than, I think we have used more than 10 million up to now. And then the research collaborators, as you can understand, all these things that I've shared cannot be done in one place. Some experiments were done at Kenya Medical Research Institute. Actually, even it is Cambridge that gave the ethical clearance. Some of the experiments were done at the University of Washington. As I've said, I went for a 3 months course. Some of the experiments were done at the University of Nairobi. Some of the experiments, actually, this is Mount Kenya University, the one where the actual research started. Some of the experiments have been done at Southern Illinois University, Edwardsville, where I was given an opportunity to see how their NMR equipment works, GCMS. It was a very advanced laboratory. I was given an opportunity to go and see how this equipment look like and how they work. So I also worked there at the Southern Illinois University, Edwardsville, and then Institute of Primate Research. That is where we did the histopathology, getting out what are the, that is an institute research in Karen. A lot of experiments have been done there. So most of the experiments have been done in this place, of course, in addition to, these are the collaborators, but also remember the main research was done at the Jomo Kenyatta Institute of Agriculture and Technology. So I think that comes to the end of my presentation, and I'm thankful for those who have listened to me. There are just 16. But this has been recorded, so I think others will be able. So this is the place where I collected my first plant in 2010. This is the area, semi-arid area in the bushes. That is where I collected my first sample. Oh, thank you very much.
Thank you so much. Thank you so much, Dr. Christine Wangia. Maybe to introduce myself, Jimmy Ngaiwate. I'm a student at Jomo Kenyatta University of Agriculture and Technology, your institution.
Yes.
I'm also studying BSc Biochemistry. Thank you so much for your wonderful presentation. Actually, it's an interesting journey and actually inspiring to us as young and upcoming researchers. As you can see that antimicrobial resistance is a very big, huge threat globally. Actually, as students, we have become motivated, and we're motivated also to come with effective approaches and innovations like yours also to curb this global health threat. Thank you so much for your wonderful presentation and your educative session like this one today. Actually, I can say that it's full of inspiration, resilience, hard work, and determination. And I think above all, it is full of passion. And that's what maybe may have driven you to reach where you are. And actually, above that, you are going to achieve your dream. Thank you so much. At this session, we are going to take it for as a question and answer session. Maybe so I welcome anyone with a question yeah, it's over to you all.
I can see some charts, but I can't read because my screen is on. I don't know what those charts, there are 7 charts. So maybe you can read out those charts and so that if I'm told to go to slide so and so, I don't come off the sharing and
Yeah, Doc, actually, most of them are congratulating you for the wonderful presentation you
okay.
had. Yeah, it's very
Okay,
nice.
I can also just tell you this is, you said you are Jimmy.
Yes, Doc,
I have done quite a lot on this plant apart from the anti-inflammatory activity. It has actually antimicrobial and antifungal activity. So if anybody is interested to further study this, I supervise a student who did antimicrobial and antifungal, and it had actually activity. So, but that is now in the lab. What I was trying to tell you that when you see some report, you can pick from there. So anybody who wants to get maybe a new medicine from the plant, you can go the antimicrobial because I've already done antimicrobial and antifungal and it showed activity in the lab. So somebody can move on and maybe get some new compound from this plant as an antimicrobial and antiactivity. Now that you people are interested in antimicrobial, it's worth studying. It's a tip for you, a student did and you got good results. Although it was not published, but.
Okay, thank you so much. We are so happy to hear that. Actually, it's an avenue for us to get into there and explore more on the plant, maybe to get some active biomolecules in the plant. Actually, I think one of us, we can just work on the plant, to come up with effective antibiotics because that's our main target as far as antimicrobial resistance is concerned.
Yes.
Maybe, I think anyone, I can just offer this chance to anyone maybe who has a question to ask before or as you continue.
Jimmy, there's one in the chat box that has just been shared.
Okay.
So maybe I can read it. Thank you so much, Dr. Wangia.
You read it for me. You read it for me.
Yeah,
okay.
It's been a pleasure. I really do appreciate. Thank you so much. And maybe to introduce Jimmy, he forgot to share that he's also the one who is heading our drug research and discovery program. He's also very motivated and like you, and he's also doing some research on H. pylori, and I'm sure this is something that we could also work on. And he's a colleague here at JQuat too. So
Sorry, you said he's doing research on what? I didn't get doing research on what?
On H. pylori, on some.
Okay, H. pylori. Okay.
Yeah. So thank you so much. It's a question from John Masharia. First, he starts by congratulating you for the wonderful work. Good job, Doc.
You.
Indeed, it has been a journey you've walked through, and your determination is encouraging to us. You're indeed a mentor, especially in our country, where such research is still not exploited. So John has one question. You said the patent has an expiry period. How long does a patent take to expire?
20 years. That is why I'm saying I'm still working on this, that before it expires. And you know, there are times, even if the patent even if you go 15 years, there's always money for renewing the patent every year. We have to do it. So also what is encouraging me that even if I take 15 years to get the drug, I'll make it for 5 years alone, and maybe I'll recover. So actually the time is going, but you know, there's no shortcut. But I have that faith that at least the remaining years, because 2016, it means 2026, 2046. Okay, what am I talking about? 2016, 2036, sorry. 2036, I have that faith that something will have come out, because why I've reached is I really need the approval PPB. If I get approval PPB, I will be able to move on. I still have some time. But passion, as I can say, passion, there are so many things like I explained to you when the Filipino challenged my work and I was delayed another 6 months. You know, you can just say, don't want this thing now, but I have that faith from the time I worked on that plant in India. I have faith this plant will come as a medicine in Kenya. I have that faith because science does not cheat. You get results, they are results. There's nothing you can do about them if you have not cooked them. They are results.
Thank you so much, Dr. Wangia, for that. I can see Kawis Yusufu has raised his hand. He's our colleague from Uganda. Kawis, you can go for it.
Thank you so much, Daniel. And also, thank you so much, Doctor, for sharing. As we all know that medicinal plants are really coming forth as a solution to most of the challenges in line with antimicrobial resistance. Doctor, I happen to be doing research in bio or green synthesis of nanoparticles from these plants. I just wanted to find out, because nanoparticles have antimicrobial activity and we are really looking forward to solutions in microbial resistance. What happens to be your take on the green synthesis of nanoparticles from plants? Thank you.
Okay, that one, Yusuf, I must admit, that area I've not gone into that direction. And I can only talk of what I've done. That one, I can't say something about that.
Okay, thank you so much.
What I know is that nanoparticles have an advantage of delivering drugs even in small quantity, but I've not gone that direction. Because really what as I've shared with you, I really want these compounds from the NMR. Because when I have this compound, then I can say this is compound so and so. That is really, I really want those compounds. So I think I'm in another direction for the time being.
Okay.
I'm sorry.
It's okay. Thank you so much. Over to you, Daniel.
Thank you so much, Yusuf. I have a question to Dr. Wangia, and I think this is something that people are thinking of. Now, as a student, as an undergraduate student, where would you advise one to start? Which route would you advise them to follow at this time, maybe before even they pursue their masters, if they're interested in, starting some research on, on this compound, on this plant compound?
I think when you do projects, I don't know, you do projects in 4th or 5th year.
Fifth year?
Do project. 5th year, yeah, you do projects. When you do projects, and that is your desire, that is where you start. Like me, the student who did the antimicrobial and antifungal, he came to me and said he would like to work on my plant, So I allowed him to do some work. I'm the one who told him how to get the plant and so forth. Actually, he did not even go into the field to look for the plant. I already had the extract with me. So he started off on a light note. Another one wanted to do gastroprotective effects. So I have 2 students who particularly who said they would like to work on my plant species. Because this one of gastroprotective also had his own reasoning. Most of the drugs that are used in arthritis have, especially the non-steroidal anti-inflammatory drugs, they irritate the GIT. So he wanted to find out, as far as safety is concerned, can this drug also be safe in the GIT? So he had ibuprofen-induced ulcers. And the medicine also worked like ranitidine. That time, ranitidine, Zantac was still being used. So you see, this plant has so many things that one can study. But what I can just tell you, as you are interested in antimicrobial, you can do the antimicrobial aspect, especially of the resistant strains. You can start in the lab. You don't start in the lab, see activity in the lab, and then during your project, that can be done during your project time. Then when you finish and you get good results, you can move on. And as I've already said, I'm looking for funding. If I get good compounds and I get funding, I will have money at my disposal, and whoever wants to work with me, of course, I'll give you an opportunity, because what I'm looking for now is funding. I told you I was going to get a National Institute of Health grant in the US. They only told me if only these compounds were NMR, they were going to give me the money. So they gave me that challenge, go back to the bench, start doing any fractionation. So I'm still having that faith that once I get these compounds and I subject them to in silico studies, the goodness of in silico studies, let me also tell you, the goodness with in silico studies, yes, I'm looking for inflammation and arthritis, but you may be looking for antimicrobial. They will also have targets for antimicrobial targets. Or even if somebody wants to do anti-diabetic, there are targets for anti-diabetic. There are so many medicinal plants that have been reported in folklore medicine for a particular activity, and yet they end up being better in another activity. So once these compounds are done, It is a matter of finding out which is the activity that you want to test. Using in silico studies, the way I showed you the molecular docking, the 2-dimension, you will be able to find out if they actually can act on those targets. And if you find out that they can act on those targets, then you can develop in vitro and in vivo studies and continue with your research. This plant has so many activities. I can tell you that whatever the interest is, you don't have to do arthritis. If your interest is something else, you can still work on that thing, because plants have synergistic effect. No wonder they are safer. This one may be offsetting the side effect of the other. So you'll find that because of the synergy, a plant can have many other activities that are good, but it's a matter of somebody studying them. What is it? So once the compounds come out, anyone can do in silico studies to determine the kind of receptors or targets that you are looking for your new drug. And then from there you move on. It's just this particular bottleneck of knowing these compounds, and we can move on in any direction using in silico studies.
Thank you so much, Dr. Wangia, for that. I'm sure you'll be in talk with Jimmy and team. I am sure, at least, because we're also planning, as I had shared, a phase 2.
Phase 2?
From around January to partners here and there. And,
you went off. I didn't get, you said phase 2, then you didn't talk again. So I don't know what you said. Just started, yeah.
Sorry. I think my...
Yeah, you went off, yeah. So you're setting phase 2 what?
Okay, maybe I can highlight, maybe you can give some explanation on that, because
okay.
Daniel, I think he has some challenges in interconnectivity.
I think my...
Hello, Dan. Or maybe I can go on. Doc,
yeah,
maybe
you can proceed.
Yeah. Maybe what Daniel was trying to say, we have a phase 2 program starting from next year, as from January up to April. It will be a first program on this program of drug research, discovery, and innovations. And that particular program, the students will receive intensive training and to learn more on drug research, innovations, and discovery. And that one will involve the tertiary level students, maybe some from colleges and also in the universities who are highly interested in the field of drug research and innovations. More particularly, on our main focus is on antimicrobial resistance. Okay. So that's what we had. So maybe as we go on, maybe we will require more assistance from experts from you, Doc, and also maybe from any other person who will be willing, and maybe we require assistance and your support on the same program that we are intending to begin early next year. Yes.
Okay, I'll be available. I'm always passionate with this thing. I can talk about it till tomorrow.
Thank you so much. We will be so much grateful. So
no
maybe,
problem.
maybe I can just also raise some questions that I maybe would like to ask. Maybe, for example, when you see that these medicinal plants actually, due to misuse and maybe subsequent extinction of this plant, especially the endangered species, who are highly good as potential and discovering new medicinal compounds. Maybe what is your take on this, and maybe the measures to protect these plants from extinction? Over to you, Doc.
Okay, that is a very good question, Jimmy. You know, I've done so much that I didn't even want to share with this. You know, this is you saw this, how small the plant species looks like, and it's a perennial creeper growing in the semi-arid area. So actually my research was delayed partly because I would wait for the rains to come. I had to wait for the rains to come. Within 3 months, go pick the plant. If I went after 4th month, I found the plant it had already dried. So this kind of thing also delayed my research because I was not able to get good quantities to do a lot of work. So what I decided, it's also another research. I'm also looking for funding. I decided to do soil analysis of the natural area, the habitat. I also did soil analysis where I stay. I have certificate of soil analysis from JQuat. And I found that that soil in the wild was similar to the soil where I stay in Kakamega County. And I can tell you when COVID came, but also people got opportunities. When you are an entrepreneur, look for opportunities. Don't just cry that COVID. When we had a lockdown from March, we decided, what are we waiting for? We have soil analysis of this plant from the semi-arid and here. We'll start off by doing domestication. So I have this thing, I have this plant. One of the JQuat magazines has my story there, I think 2019. I have a 2-acre where the plant is growing healthy. I have done in vitro and arthritic activity and this activity in this particular place. So actually I am looking for funding to domesticate, have farmers plant this as a cash crop and then develop into a medicine. So that I've already done on a 2-acre. So I had a presentation sometimes to some German farm, and on the 18th, they want to talk to me again if they can engage with me in mobilizing farmers to plant on a small scale, standardize the method of planting, then we can have it as a cash crop. So what I've done is to domesticate it. I've removed it from the wild because in the wild, the goats eat. In the wild, you have to wait until it rains. And why not have it and standardized methods of cultivation, mass cultivation. So I have cultivated this thing. I don't go to that area anymore. I just pick from my farm. So it is a domestication. The species that can be extinct, if one can do domestication, but the domestication also has its own issues. You must make sure that the soils of the two places are the same. If not the same, you enrich your soil to look like the other one, and you must test the phytocompounds, not just having the thing plant like vegetable. I've done the analysis of the roots, I've done analysis of the leaves, I've done analysis of the whole plant parts, and I've done analysis of the stems, the anti-arthritic activity in vitro. So I found that the best activity is in aerial parts. I don't need to uproot the thing. When you cut off the aerial parts, within 3 months, the plants are again ready for harvesting. So now this is like something that is in the process again of research. What is the best way to standardize that? When I tell this farmer, do ABCD, farmer in plant in plot A, and I tell farmer in plot C, do ABCD, they will have the same output. So that is what this is also the other part of research that I'm doing, domestication, getting funding to sensitize the people around to plant this, because it will be a cash crop, it will improve their livelihood. So that is also one of the ways, domestication. But before that, you have to do what I've said. You have to check the soils, because I said soil analysis, geographical locations, climatic conditions can affect the phytocompounds. So that can be, as you do this, you are also testing the phytocompounds and activity. It can be done.
That's wonderful. I'm so happy to hear that, Doc. Actually, yeah, I didn't know that we can manage to do that. But actually, thank you so much for giving more highlights on the domestication and soil analysis of the plant. Maybe soil analysis, maybe because we can see the major problem nowadays we are facing, because due to misuse of these medicinal plants, they are going to be extinct. Maybe in the near future, we may not have these plants maybe to work on. Actually, it's a very big challenge. So maybe those who may be interested to work on certain plants, maybe, the first thing is to identify the plant. Now, maybe if you cannot get the plant, automatically, that will be a very big challenge in your project. You to maybe impair your research. Actually, that's something that you need to think of as we move on, as new upcoming researchers.
Another thing which I've also tried, it is tissue culture. It's very expensive, but tissue culture is you can get mass products from a species that can be extinct. I have done tissue culture. Actually, it is the one from tissue culture is when I learned that I would do vegetative planting of this particular. But it's also expensive, Jimmy, because we've done this thing during lockdown, April, clearing the farm. By now, we have already spent 0.8 M. And we are not yet, we have not reached out, we have employed an agronomist. We have spent 0.8 M from April last year when we began clearing that farm. The research is very expensive, and that is why we have to look for funding. Without funding, it is not possible. So this all funds is just from my salary, but you see, I'm saying one day, I'll get an investor. So there's an investor that has been interested, a German investor, who listened to me sometimes back, and he's giving me an audience next week on the 18th, hoping that he will come. They do this kind of thing, sensitizing farmers. Their aim is to make the local people improve in their economy. So that if they come, if I win that, they are the ones who will come. They'll tell me who are the farmers, where we can start in the nurseries, you know. So it is them now to do the work. Once you win that, they're called develop, they will do that. So when with the funding, you can do anything. So it's just money that is delaying me. But if I got money, things would have moved very fast. It's just money. But one day it will come.
Oh, sure, Doc. I do understand. Very true. Actually, research is very expensive. And I think also maybe another challenge, maybe as you can see in Africa, maybe the major challenge, maybe there is no enough funds allocated for research. And actually, that's one thing that we would wish our governments across Africa maybe could also collaborate and set enough funds for research across Africa, maybe for purposes of research, and also increasing our capability and development in terms of research and innovation across Africa. Actually, it's a major challenge in our continent, I can say so. Maybe another question, maybe I would like to hear your advice and maybe some encouragement to the young upcoming researchers, especially students, maybe to develop, how could they develop and sustain their interest in research?
I think if you want to see far, you say you stand on an elephant. For saying that if you want to see far, you stand. You know now what I've already shared with you, and I'm very honest. I've helped so many research assistants. The ones who have worked with me, they have gone far. I've encouraged them to do their postgraduates, who are doing masters, who are doing PhD. I tell them, don't just work on this plant of mine, please do your postdoc. And two are actually doing their PhD outside the country. One is doing masters outside the country. One is doing masters here, another one finished. My research assistants have always told them to go for further studies. That is how they will be relevant. So what I'm just trying to share with you, I'm looking for funding left and right. If ever I get good funding, I will involve the students that I know. I will involve the people that I know. So just pray for me. Whoever is interested in working, doing anything on this species, if money will fall into my hands and you come, we discuss, you write a good proposal, I will guide you and you'll be part of this. You only need money. There's nothing that you can, because when the students worked on my plant, they never bought any solvents. I just gave them my solvents. So you see, once you are working with somebody who has funding or who has the resources, it is much easier. But you to do as a student is expensive. You'll have to work with somebody who is already having resources. That is how you will move. So what I'm looking for is not just for myself. I like young people. I like helping young people to move from one level to another. So if you pray that I get funding and you are interested, the funding will be there. What I'll need is write a proposal, so and so is doing this, so and so is doing this. So just pray that Bless or somebody else is money. What I'm trying to tell you that with grants, even in my research, you saw how I won the grants, the 4 million. Without money, you can't move. Okay, with money, anything can happen. Proposal, you move on with money. So it is just money. When you have money or you work to collaborate with somebody with funding, you can do and you can go very far.
Thank you so much, Doctor, for your encouragement and your willingness to support us. Actually, we are so willing to work with you, and maybe in sometimes in future, I know we will achieve a lot with you as students. We will grow together.
Yes,
thank you so much. Maybe I can just offer this to Daniel, maybe to say something as we end the session. Over to you, Daniel.
Hello, I hope you can hear me.
Yeah.
My network is having some challenge.
Can hear you.
Okay, thank you so much, Dr. Wangia, for the willingness to engage and for making time, especially on a Saturday. It's off official hours, but we really do appreciate. We try doing some of these sessions outside classes. At least Saturday and Sunday, we have some time to engage in these other activities and to empower our fellow young people. We really do appreciate that you made it today and shared with us your story. I'm sure many of us have been inspired. And the thing I love is that at least for you, you are able to do your research locally. That shows that with some support and some funding and above all the passion, we can also be able to do something locally and also in collaboration with other international partners. So mine is just a calling for all of us who have been able to attend the sessions and also those who will watch the recording. If you're passionate about it, just keep on searching. You never know, even for those outside Kenya, at least I'm sure even how you elaborated the process of patenting, I think the process is also the same in Uganda, in Tanzania, in Zambia. So they're not so much different. So I think all we just need is the passion. You keep pushing, and I'm sure we have so many people like Dr. Wangia in these other countries who are willing to support us and usher us into the research field. So thank you so much, Dr. Wangia. Maybe we can have your parting shot as we end the session. I'm really grateful.
Thank you so much. I'm actually very humbled that you asked me to talk to you because, as I said, I like working with the young people like you. I like imparting. I like making young people to be better than what they are. And if I have resources, I always share. And even advice, as I've said, most of my research assistants are not just my research assistant, they have gone far. Two are doing PhD, one is doing masters in China, one is doing masters here. I don't just let them that they are working for me. No, I tell them, yes, we are working together, but I would like you to be better, move to this direction. So I like working with young people. I was so excited to have invited me, and any other time I can talk to you. And if I get any funding, if my way opens, I'll talk to Daniel. I tell him, Daniel, something has come up. So when you are doing your projects next year, if something comes up, I'll be able to share and say, no, I got something. Whoever wants to do ABCD can do on this plan. And as I told you, this plan has many activities. They've reported antihypertensive, they have reported gastroprotective, they have reported anti-diabetic, they have reported, there's so many things that have been reported in this species. So whatever your interest is, you want to work on this species with whichever interest, you don't have to do arthritis, whichever interest, especially if it has been mentioned and have funding, I'd be able to be of help. So I'm thankful that you listen to me also and have a blessed weekend.
Thank you so much. Dr. Wange will be in touch. I'm not sure whether you still come around to JCOT nowadays due to COVID, but
no,
one day.
I'm on leave. I'm on leave till January. Actually, I'm at home. I'm on leave till January.
Yeah. So thank you so much. I think Jimmy will finish next day. So once you're around, once you come back on January, you can have a small physical meeting with Jimmy and the rest of the team members.
Yes.
That also,
it's okay.
You get also to see Jimmy and the team.
It's okay.
Yeah.
It's okay. Yes.
So thank you all. Thank you all of us for attending the session. It's been a great 2 hours. Thank you also for being accommodating despite the earlier challenges that we had. As we said, we will share the recording with us so that you can also share with your friends, and we'll upload it via YouTube so that it's accessible to everyone else who would love to learn more about the session. Dr. Wangi will also request you can also share the slide presentation so that we can also be able to share with the students too. So thank you so much, everyone. And I wish us a wonderful weekend and a great week ahead. So thank you and bye-bye. Have a wonderful weekend.
Yes, I will share the slides. I'll share the slides.
Okay, thank you. So thank you, everyone, and bye-bye.
Bye.
Bye-bye. Thank you so much, Doctor, for having you today. Actually, it is an inspirational presentation from you. And also, we would like to hear this from many other researchers across Africa, actually for encouraging students maybe to get into research, actually as students, we want to achieve maybe our aim on antimicrobial resistance. Thank you so much, everyone, for attending this session today. And we'd like to hear more from more of our speakers as we move ahead. Thank you so much. Have a wonderful weekend ahead. Bye-bye.
Okay. Bye.